Good day, grade 11 learners. Welcome to today's Life Sciences lesson. My name is Susan Wiese. This lesson is brought to you by Gauteng Department of Education in collaboration with Sibona Discovery Centre and broadcast by Pakama Research and Development. Today we are going to look at the skills needed in life sciences. Now I know you might ask yourself, why? Why are we looking at the skills in life sciences? Well, let me tell you a secret. In every paper, whether it's a test that is set at school, whether it's a national paper set by the Gauteng Department of Education or by the National Department of Education, there is a lot of marks testing the skills in life sciences. And you know what? You cannot study for that. These skills are tested to see whether you can read a graph, whether you can draw a graph. Do you know all the concepts that is associated with a scientific investigation? And because you don't know what the examiner is actually going to ask you on which topic, because it can be in any topic. It can be with reproduction, it can be with mitosis, it can be with meiosis. That's why it's so important for you to understand the skills and be able to practice and to do these skills. So this is a lot of easy marks that you can actually score in your paper. 20% of the marks in the papers are actually set on skills. So as you can see, very, very important. Before you even go into the next grade, it's very important for us to make sure that you know and you can do all the skills that is required from you as a life science learner. We're first going to look at the scientific investigations. In every question paper, whether it's paper 1 or paper 2, you will find a question on an investigation. So it's very, very important for you to be able to do and answer and phrase all the things the examiners want you to know about a scientific investigation. We're going to look first of all at variables. In planning the investigation, we need to test various possibilities or factors, one at a time, so that we can be sure which factor gives us the result. Each factor is known as a variable, and each investigation should test only one variable. Generally, a scientific investigation has an experiment and a control. And I'm sure you have seen questions in your papers, in your test, in your end of the year papers in June and first term. You might have seen these experiments and control that has been put together in one question. Now, there are 3 types of variables. And life science learners, we will never ask you, give us the definition of a variable. No, these are the application questions. So we're going to give you a scenario and then ask you to identify some of the variables. So what is important here? It's important for you to know the difference between the 3 variables that is expected of you. First of all, we're looking at the independent variable. This variable is controlled or changed by the investigator to determine what effect it has. In graphs, it's the variable that is drawn on the horizontal axis or the x-axis. So very important, remember now, the independent variable you're going to find on the horizontal axis or the x-axis. That's very important for you to remember, because if we give you a graph, and I ask you, identify the independent variable, then you must know where to look for it. And if there's a graph, remember now, it is on the x-axis, the horizontal axis of your graph. The second variable is the dependent variable. This variable is the effect of the independent variable. This is the response that is measured or monitored during the investigation. In a graph, it's the variable that is drawn on the vertical axis or the y-axis. So remember now, the dependent variable, that is the one that you are measuring. And that you're going to find on the vertical or the y-axis of a graph. So if we look at a graph, remember this is the y-axis and this is the x-axis. All right. And then the third one is the fixed variable or the constant variable. All the factors that must be controlled, fixed, kept the same when conducting an investigation. So, remember now, like I said, we will not ask you, give the definition, what is an independent variable, what's a dependent variable, what is a fixed variable or a controlled variable. But what we will do is, we will give you a scenario and then we're going to ask you to identify these variables. So for you, it's important to know, first of all, that the dependent variable is on the x-axis, the horizontal bar of the graph. The dependent variable is on the y-axis, that is the vertical axis on your graph. And then the fixed or the controlled ones are the one that we keep the same. So if you read in your introduction to an investigative question, it was done at the same time of the day every day. They used the same size of beakers. Those same things, those are your fixed variables or your controlled variables. Now, hypothesis testing. What is that? A hypothesis is an attempt to explain some event or observation using whatever information is currently available. It is an educated guess. You are predicting what is going to happen. So what is a hypothesis? It means I have seen something. And now I'm posing a question and I'm wondering, let's say we see in our school that suddenly the girls are all now, let's say, short. So now the question is, why are all the girls short? That's our question. A hypothesis would be, all the girls are short because there's now a recessive gene, or they've inherited or the food that we are eating and the water that we are drinking has a, let's say, negative effect on their growth. So you're giving a possible answer to a question that was posed. That is a hypothesis. How to state a hypothesis? Because the examiner can ask you, if this is the experiment, state a hypothesis for this experiment. How do I do that? First of all, there must be two variables. So, in which variables are those? The dependent variable and the independent variable. Secondly, state the relationship between the two variables. So you must say, if the one, let's say, is going to be higher, the other one will be also going higher, or the one is higher and the other one is lower, or whatever the case may be. But you must state both variables and you must say, what is the relationship between these two variables? And then it must be testable. You cannot have a hypothesis, but you can't test it. Because then how are you going to prove whether your hypothesis is correct or wrong? And then remember, we put the independent variable first. That's the cause. And then in your hypothesis, you put the dependent variable second. So that's very important. So these are basically the rules of how to state a hypothesis. So let's recap. There must be two variables. Which ones are they? The dependent variable and the independent variable. Which one must you put first? You must put the independent variable first and then the dependent variable. So the one that you are going to measure is the one that's coming second. And you must state the relationship between those two variables. And then your hypothesis must be testable. All right, so that's very important for you to remember that. Furthermore, a hypothesis is an idea that a person gets from observations made. So it's something that somebody has seen. It is a suggested solution to a question. So you've seen something, you've put a question, we call it a investigative question, and then you basically give a possible answer for that question, and that is a hypothesis. The hypothesis needs to be tested before one can draw any conclusion. So you're going to do an experiment to test your hypothesis, and then your results will tell you, yes, you were right, your hypothesis is correct, or no, you are incorrect, because the results is showing that what you thought the answer isn't the answer. Then with all investigative questions, there is a factor called reliability. And if you look in the question papers, the examiners ask that if you see a question on an investigation, you can expect to be asked to identify, which factors contributed to the reliability of this experiment, or how can you improve the reliability of an experiment. Now let's look. What is reliability? The idea behind reliability is that other researchers must be able to perform exactly the same experiment under the same conditions and get the same results. That is reliability. In questions which ask learners to state how the reliability of the investigation could have been improved, the correct answer could generally be the following. So remember, reliability is somebody else must go and do this experiment and get the same results than you. So it's all about the results. First of all, you can repeat the investigation, because the more time you do an investigation and you get every time the same results, then you know they are reliable. Increase the sample size. So instead of only have, let's say, 5 chickens in my experiment, I should rather have a group of 100 chickens in my experiment, because that would mean I've tested it actually more. Take many readings and use an average for your results. So you take many readings. You don't do it just today. You do it tomorrow and get a reading, and the day after that, and the week after that, and then at the end, you get an average of all your results, and that will help you to increase the reliability. Remember, to increase your results, believeness. Can somebody believe what you are saying? Increase period of investigation. So like I've said, do it not only for today and tomorrow, do it for a month or do it for a year. And then select your sample randomly. So you don't choose, deliberately certain things. You go and you do a selection, but you do it randomly. So please remember, reliability is something that you're going to be asked to identify in an investigation. And please read carefully. Sometimes they will ask you, what have the person done? So then you must go and read in your text. We will give it to you. It will be there somewhere. Just read carefully. But sometimes the examiner can also say, how can the reliability of this experiment be improved? That means it is something that you now must say they must do in future, not something that they have done already. All right. So here language is very important. So read your question very carefully. Make sure you understand what the examiner is referring to. Is the examiner referring to what was done, or is the examiner asking you what can be done? All right. So there's quite a difference between the two. Then we move to validity, the validity of an experiment. And what is validity? Validity questions how the experiment or the investigation was carried out. So this is about the method. How did they do it? Remember, reliability is about the results. Validity is about the method. Have all the factors, variables been controlled, or were they fixed, kept the same, except the variable or the factor being tested? All right, remember when we started, we said there are 3 variables. We have looked at, where do we use the independent variable and the dependent variable, stating the hypothesis. We looked at reliability, but with validity, the fixed or the controlled variables is now coming in, because you're now looking at how was this experiment done. Is the design for the investigation appropriate? You can't say, I want to test, let's say, the influence of sunlight on photosynthesis, but you never ever somewhere measure, the intensity of the sunlight. You never measure the temperature. So that means that if you look at the way that you did the experiment, there are flaws in your experiment. And that means the validity of your experiment is actually being compromised. And then when being asked about the validity of an investigation, the answer should center around the variables being kept the same. All right, so validity is all about keeping the variables the same. And that you will pick up in the text that we are giving you when we're asking you a question on an investigation. Look for the word the same. The same light, the same size of the beakers. Those things are then your fixed or your controlled variables. Then a scientific investigation is basically a hypothesis testing. All right, because somebody has seen something, has posed a question, came up with a possible answer, and now they must go and test it. So first of all, somebody has made an observation of a phenomena. What is a phenomena? It's just something that you have seen. All right, so it's a big word for something actually very simple. Then you ask a question about this phenomena. Let us here put something on the table. What can we observe that we want to test? You have seen that now in summer, the bread in the bread bin is getting covered by mold very quickly. But in winter, the bread in the bread bin can last for a week. So what is happening? Why is this? So we have observed this. So we must now have a question to say, why is bread in summer being covered by mold much faster? Does temperature play a role? Because in summer it's hot and in winter it's cold. And then we come up with a hypothesis. Through observation, identify phenomena and formulate a question. List all possible variables, all right, the factors. So now we must think about what factors are we going to list. Remember, you have dependent, independent, and fixed or control variables. And then we're going to formulate a hypothesis. So our hypothesis could be that bread in summer, or let's say bread in a hot place, is covered by mold much faster than bread in a cold place, or in summer the bread is actually being covered by mold faster than in winter. So then we must now go and investigate that. So that's just a hypothesis. We're just saying we think that's the answer. And remember, your hypothesis must be cause and effect. So you can say, in summer it's covered in mold faster because it is warmer. Then you formulate a hypothesis, a prediction that follows the criteria. Explain all aspects of an observation, be the simplest possible explanation. So no fancy words, straightforward. Be expressed in such a way that predictions can be made from it and be testable by an experiment. So your hypothesis must be testable. Then you must plan or design your investigation. And you know, in the first papers that we saw in life sciences, the examiners asked us, state the hypothesis, then we got the results, and we had to draw graphs, and that was basically it. Nowadays, they are now asking us to state or give the aim of the experiment. Why was this experiment done? Planning steps. How did this guy plan his experiment? Can you see any flaws in this experiment? So when you read this question, take your time. Take your time, read the question clearly, understand, underline the words, look for the variables. As you are going through, highlight the independent variable, highlight the dependent variable, highlight all those same things, because those are your fixed or your controlled variables. All right, so it's important for you to basically analyze the information that the examiner is giving to you. Now, when you plan or design an investigation, and remember, they can ask you to plan an investigation. They can tell you, this is the hypothesis, this is the observation that was made. Now you must go and write down how will you go and do this experiment. Identify the variable to be tested. That's the dependent variable. All right? That is the one that you're going to measure. Identify the variable to be kept constant. All right? That's your constant or your fixed variables. Identify the independent variable. And then list appropriate apparatus. So you will have to go and think, what am I going to use? What do I need to do this experiment that I would like to do? And remember, there's an experiment in every investigation, and there's a control in every investigation. In a control, that is where you actually keep everything that must be there. And in the experiment, that is where you have one factor that you're going to test. How will you collect data? So you have to think about this. Select instrument, a method that is most appropriate for collection of data in an investigation. Decide how you will record the results. If you work with humans or animals, you need to get permission and ethical clearance. So you can't just go and buy a rabbit and now you want to do experiments on this rabbit. It's not ethical. You first have to get permission to do that from the SPCA. So, and even if you want to do an investigation in school and you want to ask questions to your fellow students, you're not allowed to just do that. You have to get permission from the principal, you have to get permission from the learners themselves and maybe their parents as well. So there's a lot of thinking behind an experiment before you can actually do this investigation. All right. Now let's look at results. Recording or results or findings. Record results in the form of tables, drawings, descriptions, pictures. All right. So you now did the experiment, and then you collected data, and now you must actually put that data in a form that anybody else can read it. Identify patterns in the results. Identify and record irregular observations. All right. So you need to be a very good observer. You need to look at things in front of you. Now let's get back to our bread mold. Let's say we want to test. Is it so that in hot or warm temperatures, bread mold is growing faster than in cold temperatures? So what results will we have? What will we look at? We might put bread slices in different places. You know, same container, same size of the bread, and put them, let's say, in the bread bin where it's dark but warm in summer. We can put it in the fridge or the freezer where it's very cold, and we can put it, let's say, in a cupboard. All right, where it's not cold but also not so hot. And then after a week, we can look at the bread slice itself, and then you look at how much of the bread is actually covered by that mold. Is it 20%? Is it 30%? Is it 50%? And to be fair and to be accurate, I would actually made a little grid, all right? So that you can place that grid on your slice of bread and then count all the blocks where you have bread mold. All right, so those are the things that you need to think about when you are planning to do an investigation. And then sometimes you must reorganize your data, because if I have, let's say, bread mold, I have counted. So now what I must do, I must go and present that data that I have in such a way that other people, when they look at my results, will be able to understand what I'm actually telling them, what I've seen, what I've observed. And we can sometimes plot graphs, line graphs, bar graphs, histograms, pie charts. So there's basically 4 types of graphs we use in life sciences. We can construct flow chart, mind maps, and concept maps. So it all depends on your data. So coming back to the bread mold, you can put that in a percentage. And if you put it in a percentage, then obviously you can actually draw a bar graph, so that you can compare and easily see, on which slice of bread the mold grew the fastest. Conclusion, when do we write a conclusion? And please, examiners nowadays, do ask us, give the conclusion for this experiment. So you must be able to look at the data and write the conclusion. And remember now, the conclusion must speak to your aim and your hypothesis, because you're now actually saying in your conclusion, my hypothesis that I started off with there in the beginning, is it right or is it wrong? That is what the conclusion is all about. Explain the relationship between the variables. Remember, in your hypothesis, you had the 2 variables. So now you must explain, how is this 2 now still relating to one another after you have now done the experiment. How and when the dependent variable changes over time, or concentration changes, pH, light, carbon dioxide. So it depends on what you have investigated during the experiment. Identify and explain trends, patterns from graphs. So you need to look at your graphs if you have a graph, and you have to, in your conclusion, say, what do I see in my graph? All right. Flowcharts, descriptions, whatever you have, you need to maybe do calculations if it's necessary, and if your data is of such a way, such a nature. Explain your data. That is what a conclusion is all about. You must explain the data. Now, in the exam papers, we normally give you the data. We give you the scenario. We tell you what have they seen, and then what have they decided to do, how did they do the experiment. We give you the results, and then we will ask you all these questions. So you must be able to explain the data in your conclusion. And then suggest specific changes that could improve the apparatus and the technique. That is more when you are doing the experiment yourself. You have to reflect. When you're done, you have to reflect to say, you know what, maybe if I have used more slices of bread, maybe if I have used more places where I've put the bread, I might have gotten a better result. That's the type of reflections that you have to do after you've done a scientific investigation. And then compare your data. Look for similarities, look for differences. Examples of a scientific investigation. Now that we know what all the concepts are that is related to a scientific investigation. Observation. A researcher observed that the potted plants he covered with a black plastic bag did not grow. All right, so that's something he saw. He saw that if I put a plastic bag over my plants, they don't grow. What is his hypothesis? Because remember now, he has seen that. Now his hypothesis is sunlight is essential for plant growth. That is his hypothesis. So he's saying that's the answer to what he has seen. What variables are there? In planning the investigation, he set up 2 sets of 50 plants as follows. In the experiment, he had 50 plants placed in a greenhouse. Now that is where the conditions are perfect for plants to grow. And then his control was 50 plants placed in an identical greenhouse, blackened so that no light could enter. Right, so that's his control. There he has taken out the sunlight. And then what is very important is the experiment and control differ in only one factor. You cannot test more than one thing at a time, because then how will you know which factor is actually responsible for your results? And that's your variables. That is, in the experiment, plants were given light and the control plants were not exposed to light. All right. So this is just a simple way of applying now our knowledge that we have gained from all the concepts that's related to a scientific investigation. Validity. The plants in both the experiment and control received the same soil, amount of water, temperature, etc. In other words, they were given the same requirements. Remember what I said? Validity is all about the steps, the how did you do this experiment. And when you get a question like this, look for the words. They had the same soil. All right, the same soil. What does the same soil mean? So exactly, same amount, same type with humus and whatever, amount of water. So when he watered the plants, they all had exactly the same. Let's say 100 milliliter water given at the same time. All right, all those things that are this that you do the same for everything in your experiment. That is the things that actually improves the validity of your experiment. He measured the growth of the plants over a period of time in the experiment and in the control. All right, so he must have thought about, how am I going to measure how fast or how tall are these plants growing? So he had a measuring stick, and from where to where do you measure? Do you start measuring from the tip to the soil or just the last part every time? So that's things that you need to plan for in your planning. He found that those plants placed in sunlight, remember that is now the experiment, the ones that were in the greenhouse, grew. He found that those plants placed in darkness, the control did not grow. All right, so that was basically what this guy saw. Now he gets to a conclusion. He concluded that his hypothesis was correct for the type of plant that he has investigated. All right, so this is just a typical example of a scientific investigation, put very simply. Now let's look at an activity on a scientific investigation. Two students decided to investigate the effect of different concentrations of sulfur dioxide on the germination of oat seeds. They set up trays of germinating seeds under a clear plastic cover along with 5 different concentrations of sodium disulfate solution. Sodium disulfate breaks down to release the sulfur dioxide into the atmosphere. All right, so this is what the students did. Now, there's the results, and as you can see it, they've put it nicely in a table. In the column headings, and this is a column, this is column 1, that's column 2, that's column 3. In column 1, they have the concentration of the sodium disulfate. And then in column 2, they have the number of seeds germinated out of 20, all right? And then he worked out the percentage of the germination. So he also did some calculations here, or the two of them did some calculations. The table showed the results after 1 week, all right? So after 1 week, this is what he saw. And very important, while we're looking at this table, can you see that there is a heading for this table? Plus, there are column headings, and this is a column heading. This row is your column headings. And remember, in life sciences, you never ever put the unit, in this case, the percentage, in the cells itself. So we don't write here 0.00%, and then 0.05%. All right. In life sciences, we put it actually at the on the top, all right, in the column heading. So that's very important for you guys, that you put it in the column heading. So you must notice this. All right. Now let's look at the questions. Formulate a possible hypothesis for this investigation. All right. So now please remember, what did we see? It was two guys and they wanted to look at the germination of the seeds and under the influence. All right. So what could be their hypothesis? State the independent variable in this investigation. All right. Independent. Remember, dependent is the one that you measure. So what is the independent variable? Name two factors which might affect seed germination and which must be kept constant. All right. So that is now this is going to influence your validity. And then why was the investigation repeated 5 times at each concentration? Why didn't they just do it once? But why 5 times? All right. Remember, it's all about results. So I'm thinking here we're looking at reliability. Let's look at our answers. Remember, first one, our hypothesis. An increase or a decrease in the concentration of sodium disulfate will result in an increase or a decrease in the percentage germination of the oat seed. Right, that's a possible answer for his hypothesis. Or an increase or a decrease in the concentration of the sodium disulfate will have no effect on the percentage germination of oat seed. So you see, there are more than one hypothesis for this investigation, because you are giving a possible answer on a question. All right. You don't know yet whether what you are thinking the answer is it correct or is it incorrect. The concentration of the sodium disulfate, that was our independent variable. All right, because you gave them the same. What was kept the same? Temperature and water. So that would be our fixed variables, and that will help us to have reliable or actually valid results. Valid results. And then average estimate increases reliability. All right, remember the question was, why did they do it 5 times? And that is so that they can work out an average, and that will increase the reliability of their results. Now we're going to look at the different types of graphs that you must be able to read and construct in life sciences. And normally, if they ask you to construct a graph, you get 6 marks, or 6 marks is awarded for that question. So what is important? You must be able to do it. It's 6 easy marks that you can put in your pocket if you know how to do it. You cannot study for it, because you don't know what they're going to give you. You don't know which values they're going to give you. So this is a skill that you must practice, and you must make sure that you can do it, because it means 6 marks in your pocket without studying for it. Now let's look at line graphs. How do we draw a line graph? First of all, step 1, identify the dependent and the independent variables from the information you are giving, usually in table format. Now, remember what I said. We will not ask you, give us the definition of an independent variable and a dependent variable. But we're going to ask you to apply your knowledge. Now, what are we saying here? Identify the dependent and the independent variable. You must know by now. The dependent variable is the one that are being measured. All right. And then the other one will be the independent variable. Independent, this is the variable that the investigator can change. The dependent variable changes as the independent variable changes. For instance, the time in hours in this example that we have looked at. Dependent, this is the variable or factor that is being measured. All right. So if you can determine from the information given to you, what have they measured or what have they counted, then you have already identified the dependent variable. the temperature in degrees Celsius in this example. All right, so they're looking at temperature. Example of data that are used for line graphs. All right, like we said, you can get a table like this one. Left hand side you have time in hours, right hand side you have temperature. And remember what I said, in life sciences we do not include the unit in the cells. If you look here in the right column, where is degrees Celsius placed? There. On top, it's part of your column heading. So you don't write here in every cell 16 degrees Celsius, 24 degrees Celsius. No. In life sciences, we include the unit in the column heading on the top. All right, so that's important for you also to remember. The independent variable is usually given in the first column of the table. All right, this is a hint. The hint is saying if the scientist who did the experiment, if that person wrote his data in the correct format in a table, then normally in the first column you will find the independent variable. So this is the independent variable. And what was measured in this case? The temperature was measured, and therefore this is the dependent variable. All right, so that's something that you must remember. If I'm not sure, look, and there's a table given, look at the table. And the first column is normally the independent variable. So this is a hint for you that you must remember. How to draw a line graph? How do we do this? How do we draw a line graph? I know you think, oh, it's easy. I must just have two axes, a y-axis and an x-axis, and then I'm going to draw it. No, there's certain things that you must make sure that you do correctly. Draw a set of axes and label the x and the y-axis. The dependent variable goes on the y-axis and the independent variable on the x-axis. All right, so remember that. That's something that you must remember. The examiner is not going to tell you where must you put what. You must know this. And that's why I say this is a skill. If you know it, then you're going to score marks. There's our set of axes. Remember, this is our x-axis and this is our y-axis. All right. And we always label our axes. So you can't just have a line. You must make sure that your line has got a name. In this case, time, and then there's the unit in brackets. The same with your y-axis. The name of this axis is temperature. So this is where you're going to read off temperature. And then unit there in brackets. All right? And as you can see here, it's also a scale. And the scale here is 2, 4, 6, 8, 10, 12, 14, 16. So there's a difference between each one of them of 2. While on your y-axis, can you see, there's your scale. There's a 5, there's a 10, and a 15, and a 20, and so forth. So that's the first step when you draw a line graph. What do I need for this? A ruler. You cannot go into the exam hall without a ruler in life sciences, because you must be able to draw these lines. All right, so you must be able to draw your set of axes. The independent variable is usually plotted on the x-axis, all right, and the independent one here on the y-axis. Choose a scale for the x and the y-axis, and this is where many learners lose marks. Make sure that the scale includes the highest numbers in the table for each of the variables. Do not use the values for the y-axis directly from the table unless they have regular intervals. So don't let us catch you out. Make sure when you look at the values and you see the values are, let's say, it's 0 and 5 and then 7 and then 11, then it means you cannot use that as your scale, because the difference is not kept constant. Then what you do, you look at the smallest number and you look at the biggest number. And then let's say it goes from 0 to 20. Then you choose, I'm going to count in what? I'm going to count in twos or in ones if you have enough space to draw a big graph, or in fives, all right, so that I can fit all those values that they are giving to me onto my axis. So this is step 3. All right, you're going to choose a scale. For example, let's look at this one. This was given, and this is an axis drawn by a learner. Look at this scale: 0, 5, 9, 14. From 0 to 5, what is the difference? It's 5. From 5 to 9 is 4. From 9 to 14 is 4. From 13 to 17 is 4. And then 17 to 20 is, can you see, there's no same intervals. And therefore, this is wrong. And you're going to lose your mark. All right? Something very easy to get marks with, but also, if you're not careful, it's a place where you can easily lose those easy marks. Let's look at step 4. Place a dot at the point where the two values for each result intersect or meet. What do we mean? This is what we mean. Let's say you have to plot that at time 5 hours, the temperature was 25 degrees. Please take your ruler. You don't have to draw little dotted lines like this. All right, you don't have to draw these lines here. You can draw in pencil so that you can later erase them. But where they meet, this one goes straight up and this one horizontally. Where they meet, that is where you put your dot or you plot the point. It's important that you use your ruler. Don't have skew dots, because we're not going to award you marks. Make sure that you plot these values that we are giving to you in your paper very, very carefully. And in this case, the first one at 0 hours, can you see the temperature was, if I must say, it looks like almost 16, 17 degrees. All right. Therefore, it's important that your scale here must not be too small, because then you're going to battle. It would be much easier, 5 will work, but it would be much easier if it was 1, 1, 1. Let's say from 0, then 1, then 2, then 3, then 4, then 5. Then you don't have to figure out, in between, where's 17, where's 18. All right, so that's very, very important. In the example, the point where 5 hours and 24 degrees Celsius intersect on the graph is indicated by the second dot on the graph, like we've done here. Plot all the points using the information in the table. All right, so there all the points were plotted, as you can see, there and there and there and there. All right, all the points were plotted coming from the table. Then, after that, what do? Join the dots using a ruler. I want to stress this. You must use a ruler, not freehand. So you don't just, go from dot to dot to dot. No, you need to take a ruler and draw a line from point number 1 to point number 2, and from 2 to 3, until all the dots have been joined in sequence. All right, so you need to take your ruler and then join it. Please remember, let's say our first point was here and we didn't have this one. Then where must your line start? Here. From here to there, from there to there. And you're not drawing a line from there to 0. All right, that's also important in life sciences. We don't do that, so you must not do something like that. Then you're going to lose your marks. So you start joining the dots where the dots start. Don't extend that line to this 0 point here. And then step 6, give the graph a heading or a caption. I promise you, you get a mark, sometimes 2 marks, for the heading of the graph or the caption. So don't forget this. Once you've drawn and you've done your graph, remember to go and write a heading on top. The heading or caption should include both variables. Very important for you to get a mark, both variables. Which ones are they? The independent variable and the dependent variable. So both of those variables must be included in the heading of your graph. In this case, both air temperature and the time period of 24 hours must be mentioned in the heading. Alright, so it must be there. A line graph showing the air temperature measured over the period of 24 hours. That is a complete heading for this graph. Alright, very important. If the graph has two lines on it, then you should draw a key to show what the different lines represent. For example, the water absorption per hour of beetroot and carrot plants. Alright, because it's two different sets of data. The one set is for the beetroot and the other set is for the carrot, which means if you have two solid lines, how must the examiner know, or the marker rather, which one is which? So you must have a little key here on the side indicating to us. The dotted lines is that for the beetroot and the solid line is that of the carrot. Or if you have color pens, you can have a red line and a blue line. But then here you draw a red line and a blue line so that we can easily see that's the red one and the carrot one. So in this case, it's basically then that one. All right, and then the blue one, let's say for the beetroot. So it's important for you to give us a key the minute you have more than one set of data on a graph. All right, let us now draw a line graph. Let's see if you remember all the rules drawing a line graph. A person took part in an experiment on the eye's response to light. A lamp was placed at 7 different positions from the person's face. The diameter of the person's pupil was measured at each position. The table below shows the diameter of the person's pupil when the light was placed at various distances from the person's face. All right, then we have their table and they give us the data and they say draw a line graph to represent the information given in the table. Now, first look at the table. Remember, when looking at the table, there are certain things that you must be able to identify first. First of all, what did we say? Where is the independent variable normally in a table? We said in the first column. So this is our independent variable. And then after that will follow your dependent variable. And I said, what is the dependent variable? The one that is measured. In this case, can you see? This is how they measured it in millimeters. And that's the distance from where the person or where the lamp was placed. So before you even go to your graph, you have now already identified the independent variable and the dependent variable. And you know where does what go. Remember what we said. The independent variable goes where? It goes on the x-axis. The dependent variable, the one that you're measuring or that was measured, goes where? On the y-axis. All right, now you go and you draw a graph. A line graph indicating the diameter of the pupil of a person if a lamp is placed at different distances from the person. All right, so we've put the caption. We know it's there. We're going to get 2 marks. Because why? We have included both variables. Now, and now we have drawn our axes. We are now labeling our axes. We're telling the marker which one is which. In this case, distance of the lamp in meter is the independent variable, so it goes on the x-axis. Remember, this is your x-axis. And then the dependent variable is going on your y-axis. That is your y-axis. All right, and that is the one that is measured. There's our scale, very easy. The one for the x-axis is 1, 2, 3, 4, 5, 6, 7. And then there is the one for the diameter. Can you see I'm counting in 0.3? So it's 0. I mean in 0.8. So 0.8 is the first one. Then I have 1.6, 2.4, 3.2, 4, and then lastly 4.8. All right. So I'm not using what they have put in the table. I have chosen my own scale because otherwise I'm going to lose a mark for scaling because the values that they have is not exactly the same difference every time. And then we plot the points. All right, there's the first one, the second one, the third one, and number 4, number 5, number 6, and number 7. All right, so my points are plotted. And now what do? I take my free hand and I just connect all the dots. No, I said you do what? You take your ruler. You must take your ruler and then you connect all of them. All right. Now we're looking at the second type of graphs, and that's your bar graph. If we look at bar graphs, first of all, how to draw a bar graph? You look for the variables. Independent variable, dependent variable in the aim or in the heading of the table. You write the heading or the caption, all right? Remember we said both variables must be in your heading. The heading or caption should include both variables, otherwise you're going to lose your mark. Draw a y-axis and an x-axis using a ruler. Write the dependent variable as a label on the y-axis and the independent variable as a label on the x-axis. Remember the units. All right, if you don't include the units, you're going to lose mark. So that the units are that millimeter or meter or degrees Celsius, something like that. Ensure that your scale lines on your y-axis are perfectly spaced. Use a ruler. Now, if I can give you a hint. When you're writing, you have lined paper. There's lines drawn on your paper. All right, you don't get paper to do your graph on. You must do it in your answer book. Use the lines for the points where you're going to put the scale of your y-axis, because you know that they are equally spaced. All right, so you can't make a mistake there. But then on the x-axis, please make sure that you use your ruler so that your intervals are nicely spaced, and they must be exactly the difference between them must be exactly the same. All right, that's very, very important. The scale on the y-axis should be even intervals. The bars should be of equal width, and the spaces between the bars should be also of equal width. This is very important. We're looking at this, and if it's not correct, you are definitely going to lose a mark. So what do we say? When you draw the bars, they must be, if your bars, and I always say the easiest way to go is make the scale on your x-axis, make it 1 centimeter. So every 1 centimeter, you actually have a line. All right? So if I do this, every 1 centimeter, let's say this is 1, 2, 3, 4, all right? And now if I must draw my bars, use 1 centimeter as the width of your bar. So there will be the first bar. Oh, I'm now drawing it a little bit skew. Leave one open, and then you draw the next one. Because then you're adhering to the bars must be the same width, plus the distance or the spaces between them must also be the same. Then you know what, my bars are 1 centimeter, the width in between them is 1 centimeter, and you know you cannot go wrong. All right, that's very important. How to draw a bar graph. Draw bars to represent the number of organisms found at each of the points along the river. Bars should not touch one another, as the points along the river have no direct relationship with each other. All right. Remember, if we tell you a bar graph, then you must know it's bars with spaces in between. Otherwise, if they're supposed to touch one another, we call that a histogram. And then the question will say, draw a histogram. In life sciences, we actually tell you guys what type of graph must you draw? We will say, draw a line graph, draw a bar graph, draw a histogram, draw a pie chart. So we're actually helping you. So please read. Don't be in such a hurry that you know what you've misread, that it's a line graph. We're giving 1 mark for doing the right graph. So by doing a line graph, even if your line graph is not correct, but it's a line graph, you get 1 mark. All right, so please be careful when you read your questions and you actually answer your questions. And remember now, in a bar graph, they don't touch. Make them 1 centimeter, 1 centimeter open, and then your next bar is another centimeter. There's an example of a bar graph. The bars must be of the same width and must be of the same distance apart from each other. Bars can be presented vertically or horizontally. My advice is draw them vertically, up, straight up. Don't draw them like this. All right, draw them vertically. Okay. As you can see here, the bars, that's a bar, that's a bar, there's another one there. The width is exactly the same. And then the distance between them from here to there is exactly the same. All right, but like I said, I've seen this. My advice is keep everything 1 centimeter. 1 centimeter bar, 1 centimeter open, another 1 centimeter bar. Then you know you're not going wrong and you're getting confused of which was being now which. 1 centimeter bar, 1 centimeter open, and then you know you're going to get that mark for your bars being drawn correctly. There's an activity. Let's quickly look at this one. Study the table below, which shows information on some of the earthquake-generated tsunamis which occurred between 1918 and 2003. And then there you can see they look at countries, different countries. There's the years given, all right, and then the countries are Philippines and India and Turkey and Pakistan and so forth. Magnitude on the Richter scale and number of deaths. So they give us a lot of information in this table. Now they say, draw a bar graph to show the number of people killed by tsunamis between 1953 and 2003 in Turkey, Pakistan, Mexico, Japan, and Algeria. The examiner is trying to catch you out. He's not saying draw a bar graph of the information. He's telling you exactly which ones you must draw. Can you see you must draw from where? From 1953 to 2003. So let's go to our table. From 1953, so you must start here, up to 2003. So you must draw this data. Not everything from 1918, only from 2003, from 1953 to 2003. Must you draw all the countries? No. Which countries must you draw? You must only look at Turkey. Where is Turkey? So you must use that one. Pakistan, that one. Mexico, Japan, and Algeria. And that is what you must draw. So you're not going to even look at those ones there. And remember, this one is also, you mustn't use this one. You mustn't draw for the Philippines. Okay, so you mustn't draw for the Philippines. Can you see how many bars must you have? 1, 2, 3, 4, 5. Only 5 of those countries. So the minute we mark and we see you've got 6 bars, you're going to lose marks. Or you have more than 6 bars. You only must have 5 bars because the examiner told you exactly what you must draw. And there's our graph. Let's quickly look at our graph. There's the heading. Can you see you must indicate from 1953 to 2003 and in which countries? Only those 5. Turkey, Pakistan, Mexico, Japan, and Algeria. All right. And then look at the bars. Can you see the width of the bars is exactly the same? The distance between them is exactly the same. There is the label for the y-axis Now I'm lying for the x-axis, the country, and then there you have all the numbers of people that have died. And here should have been what? What's missing here? The label. Because how do we know what that number stands for? So there should have been a label to say that's the number of people that have died. All right. Now let's look at how do we mark this. You have now done this, and how do we mark this? First of all, if your heading is complete and correct, you get 1 mark. Can you see? 1 mark if this is complete. What do we mean by complete? Both variables. There must be an independent variable and there must be a dependent variable. Type of graph, is it a bar graph? The minute your bars are touching one another, it's no longer a bar graph. Then you've drawn a histogram, and then you lose a mark. So, all right, and look at what's the criteria. Bars not connecting, so they mustn't touch one another. Same width and spaces between the bars, the same. All right, so the bars must be the same width, and then the spaces between them must be the same. If it's not like that, you are losing a mark. Is the scale on the y-axis correct? Yes, there's a nice scale. It was counted in 500s. It's 0, 500, 1000, and so forth. Label of both axes correct. In this case, I think it was cut off here, but because there's a label, but this label is missing here. All right. Plotting, if you have plotted 4 bars correctly. So we're looking at where is your lines, you get 1 mark. If you have plotted all the bars correctly, you can actually get 2 marks. And if I count, it's 1, 2, 3, 4, 5, 6. 6 marks. So it's 6 easy marks. But you must do it correctly. All right, let's move on. Sorry. Let's look at a histogram. Remember what I said. A histogram is like a bar graph, but the bars are touching. That's a histogram. How to draw a histogram? A histogram is drawn in exactly the same way as a bar graph. So you have a heading, you have a y-axis, you have an x-axis, you're going to have bars, but remember now the difference. The only difference is that a histogram is used when the independent variable is groups of information along a continuous scale. Note that in a histogram, the bars are drawn without any spaces between them. This is very important. That's the difference. A bar graph, bars with spaces. A histogram, bars with no spaces. So they must be touching one another. All right, so you must know the difference. Example of a histogram, a table representing number of pupils in each percentage range. All right, can you see? That is where you have a range in percentage and then the number of pupils. So it's not an exact point. It's not, let's say, the range is from 0 to 19. It's not at 8% or 10% or 11%. It's a range that is given. How do we do this? Let's look at our answer. If you look here, you can clearly see the bars are the same width, all right, but there's no spaces in between them. They are touching one another. That is very important. This is the range in percentage, so this is the label. The scale here could have been 0 to 19, 20 to 39, and so forth. Number of pupils, we get this from here. From 0, I'm lying, from 0 to 19, there was actually nobody here. So there should actually be, we can draw a line there to say this is 0 to 19. Then this would be 20 to 39, and then 40 to 59. Right. So that is how we draw a histogram. Nothing different from a bar graph, except for the fact that now the bars are touching one another. There's the heading, number of pupils in each percentage range. And then there's an activity on a histogram. Translate the following information showing the rainfall in millimeters per month for Tshwane in 2004 into a histogram. All right. So now you can see, what have they measured? They have measured the rainfall. All right. And what is important is, because this was measured, then you must know that this is what? This is your dependent variable. And what did we say? If they give you a table, where is the independent variable? The independent variable is normally the first column of the table. So this is our independent variable. Okay. Now let's look. First of all, there is our heading, histogram, to show the rainfall per month for Tshwane in 2004. And there is the histogram drawn. Now let's look. There is the labeling for the x-axis. There is the labeling for the y-axis. Can you see the units are included? Months, and there's millimeters. There are the bars that are drawn. If you look here, you'll see July, August. There's nothing drawn here. Why? If we look at our table, July, August, there was no rainfall. All right. So you can have a line on the x-axis because that is where 0 is. And then you can carry on, and there is the names of basically all the bars, in the sense of this was for January, this is for February, this is for March, and so forth. All right. So for this one, if I look at this histogram, you can actually get 6 marks because everything here is correctly done. Now we're moving to pie charts. Pie charts is a little bit more difficult than a line graph, a histogram, and a bar graph. Why do I say that? Because you have to do calculations here. Before you go into drawing the pie chart, you first have to calculate the degrees of every sector of the pie chart. How to draw a pie chart? A chart shows data as a percentage or as a relative proportion of the total of a circle. All right, so it is a little bit more difficult. You draw a pie chart by dividing a circle into sectors or slices. You can actually think like it's like a pizza. To represent each item as a percentage of the whole. A complete circle represents the whole of 100%, and a half circle represents 50%. All right, so that is how you draw a pie chart. A pie chart is a circle, all right, and in that circle, you are actually going to divide that circle into segments, and each segment is going to represent a portion of the data that's actually been given to you. All right, how to draw a pie chart? To draw a pie chart, we need to represent each part of the data as a proportion of 360 degrees. Why? Because there are 360 degrees in a circle. All right. Add all the data in the table together. In this case, you will add all the columns in the CO2 column together. So now let's look at our table. We have a table, and it says the table below shows the percentage of carbon dioxide emitted by different sectors in a certain city in South Africa. And then there's the culprits. It's transport, residential, industrial, commercial, and other. And they say that is the percentage emissions. All right. Now this one is easy because if you add all these up, all these numbers up, if you add 25, 75, 15, 28, and 5, you actually get 100. All right. So now you must go and divide your circle to represent that values. All right. How do I do that? Do the following calculations to calculate the angles of each sector. Each value is divided by the total times 360 to give you the number of degrees. This you must remember, because this formula is not going to be given to you. You must know it. You must know that each value, I'm going to divide, let's say 25 divided by that 100 times 360. Why 360? Because that's how many degrees there is in a circle, and that answer is going to give me the degrees that sector must be to represent the 25. Now let's look at our calculations. There's transport, it's 25 out of 100. So multiply by 360 degrees, give you 90 degrees. Okay, so transport must be 90 degrees sector in our circle. Residential, 27 divided by 100 multiplied by 360 degrees gives you 97.2 degrees. Industrial, 15 divided by 100 times 360 gives you 54 degrees. Commercial, 28 divided by 100 multiplied by 360 give you 100.8 degrees. And then 5 divided by 100 multiplied by 360 degrees gives you 18 degrees. Now, you cannot go and measure 97.2 and 100.2. All right. So how are you going to do? This one, I will actually say, I'm going to use this one as 101 and this one as 97. All right. Because when you add this up, it must give you 360. So you can check yourself in the exam or in a test. Once you've done your calculations, add up all the degrees. If it doesn't give you 360, then you know you've made a mistake somewhere. Then go back and recalculate. All right? A compass must be used to draw the circle. Please don't think that, you know what, I can draw a perfectly round circle. You can't. All right? And a protractor must be used to construct the accurate angles for each sector or slice. And that's why we are saying to you that as part of your life science tools that you must have a compass and you must have a protractor and you must have a calculator. Because otherwise, how are you going to do those calculations we've just looked at? All right, so that's very, very important. And I know you think, oh, but how do they mark this? We actually make a transparency of the correct answer. And then we place that transparency on your pie chart to see whether the sectors is correct. So you have to work here very, very correctly, precisely. Now let's look at this one. Use the values you calculated. Remember the heading. Label each sector and write in the angles. If you are not labeling each sector, you should make a key which explains what each sector represents. All right, so where do you start? Where do I start with this? First, I draw a circle. All right? And then if I were you, I will draw for myself in the middle of my circle a straight line through it, so that I can put my protractor, my compass neatly on it, and I can measure the degrees. And when you're done with this, you move it and you measure the next one, and you measure the next one. All right. And I know you're doing this in maths or mathematical literacy. So it's not something new to you. But if you don't have the tools, you cannot draw a perfect pie chart and you're going to lose your marks. All right, it can also look something like this. So you don't have to write on here. You can have a key like this one here. This one is color-coded. Transport is the green one. So there's the transport. And I think if I remember correctly, this is 90 degrees. There's other residential, industrial, and commercial. All right. So this is how we actually draw a pie chart. You must be able to do this. 6 to 8 marks, because remember now you have those calculations that we must also award marks to. So please make sure that you are able to do this. How we mark a pie chart? Correct type of graph. So if you've drawn a pie chart but it's incorrect, you will get 1 mark. Title of the graph. Remember what must be there. Your dependent variable and your independent variable. Calculations. Can you see? You get 2 marks if your calculations are correct. If you've done everything correct, you get 2 marks for your calculations. If one of them is incorrect, you're losing a mark. And then we're looking at, can you see the correct proportion for each labeled sector? Like I said, we place the transparency on top of yours and we see whether your lines is exactly where our lines are. And if it's not, you're going to lose a mark. A pie chart activity. A group of learners conducted an investigation to determine the average amount of water used by a household. They collected data on water use from 5 families in their neighborhood. The results are shown below. Right, there is our independent variable. All right, this is independent. This is dependent variable because this is the one that was measured. We have toilet flushing, baths and showers, gardening, washing clothes and dishes, cooking and drinking. Draw a pie chart to represent the data on the table. Show all calculations. All right, we actually remind you in the paper that you must show your calculations because you're getting marks for it. So please make sure that you show them. Don't do it here somewhere on your paper. You must show it in your script so that we can actually mark them and give you the marks. Even if your calculations were correct and your pie chart is 100% correct, but we don't see the calculations, we don't give you those marks. So it's important to show your calculations. All right, let us look. There's all the calculations done. Remember, you multiply by 360 for the degrees, and then when you are done, please add this up and see whether you get 360 degrees. Right, that's important. There's the answer. Can you see here we are also using a key. It's much easier. And in this case, it was also color-coded. So toilet flushing, baths and showers, gardening. Here's gardening. This gray one. And washing clothes and dishes, cooking and drinking. All right, cooking and drinking is actually the least where our water is being used. Is in cooking and drinking. All right, there's a lot in gardening. And there's a lot in toilet flushing. All right, so that's where we are actually wasting some of our water. With diagrams, I just want to tell you the following. If we ask you to draw something, a sperm cell, a flower, a seed, the brain, whatever we ask you to draw, make sure you have a caption. Whenever you draw something, whether it's a table, whether it's a graph, whether it's a diagram, there must be a heading, a caption. Use pencil to draw clear, smooth lines, label in ink. All right, so your labeling is in ink. The diagram must be large enough to see all the structures that are in the diagram. So don't make it 5 centimeters, make it half a page. Diagram must be positioned in the center of the page. Diagram must be 2-dimensional, show length and width only, and no shading. We don't color in our diagrams. That's important for you to remember. So if you look here, draw lines with a ruler. Can you see straight lines? Your label lines must be straight with a ruler. Lines must not cross each other. All right, so you cannot say, this one is actually that one there. All right, that you're not allowed to have lines that is crossing one another. Lines must not have arrows at the end, so we don't put arrows here. Sorry for that. We don't put an arrow here or there. I know in some textbooks they have arrows. All right, but please, when you do it, do the correct thing. No arrows. Lines must touch the structure that is labeled. Yes. So let's say you want to say this is a red blood cell, then you must draw from there. You can't, start here and it's not touching anything. So it must touch. Lines on one side of the diagram if they are few. Otherwise, you go right and you go left. Labels must be aligned neatly, preferably one label below the other one. All right, so make it neat. And then tables. I've actually indicated tables already to you guys when we looked at the tables that was given to us. How to draw a table. Remember, your columns must have headings like this one here. You must draw the table. You cannot do something like that. That's not a table. All right, please draw a neat table. Have, the outside, have the inside lines. So make sure that your table is neat. What must be there? A heading. So here must be a heading. You must say, what is this? Then you must have a column heading, like I've indicated here. Humans on that side is a column heading. African apes on this side is a column heading. And if you look here, in this case, it is looking at differences. So please make sure that you compare what you have on this side at number 1 with this side number 1. You cannot have mode of walking, let's say here, and then this one mode of walking was standing here at number 5. All right. So what you have on the left-hand side in the first line or row must be on the right-hand side, that first line of row. All right, so that's important that you make sure that you keep your things together. Caption in each column. Draw rows and columns with a ruler, and the same factor is compared in each row. All right, that is very important. Oh, okay. All right. Percentage differences in calculations. This is the last one that we need to do. Calculating percentage differences. And please, you don't find this in your textbook. You have to make a note of this. When you have to calculate the percentage increase or decrease in data, you divide the difference between the values by the starting value. For example, the following table is a summary of the rhino poaching incidents for South Africa from 2000 to 2010. There we have a table. They show us how many rhinos were killed in Northwest and Eastern Cape in those years. All right, and they also give us the table. And I think we are going to have a 2-minute break now, just for me to quickly have some water to drink. So in 2 minutes' time, we're going to start again with how to calculate percentage, because it's not in your textbook, but the examiners expect you to do it. So don't go away. You can send us your messages on 064-748-8913. That is your WhatsApp messages. Don't call that number, just send a WhatsApp message if there's anything that was not clear to you. So in 2 minutes' time, we will resume, and then we'll quickly look at how to calculate percentage increase.
3, 2, 1, you're live.
Do you know that you are different? Do you know that your fingerprint is different? It's different from your peers, from the people that you are studying with. So do not compare yourself with your peers. Do not compare your results with what they're getting, because you are different. You have your own talents and skills, and also you have your own gifts. Embrace what you can do. Always put in the work to make sure that your results become better than what they were yesterday. But do not compare yourself, because when you compare yourself, you do not know what another person has, and they do not have what you have. So believe in yourself. You are different, and keep on putting the work.
Attitude of gratitude. What does that mean? An attitude of gratitude is the quality of being thankful. It's the quality of giving thanks. Ubonga, ulebuha. It's to be grateful. You and I have so much to be grateful for. You and I have parents who make sacrifices day and night for us to be at school. You and I have people who believe in us when we don't believe in ourselves, who love us when we don't love ourselves, who invest in us day and time again. Whether it's a teacher, it's a principal, it's a driver, it's a guardian, we have so much to be grateful for. And so I challenge you, young person, be thankful. Ubonge, lebuha. Be grateful each and every single day of your life. Strive to display an attitude of gratitude.
All right, let's quickly look at percentage difference calculations. And like I've said, this is not in your textbook. So please make sure that you take note of the formula you have to use to calculate the percentage increase when something happens. That's very important. And examiners love to ask a question on percentage increase in our papers. So when you have to calculate the percentage increase or decrease in data, you divide the difference between the values by the starting value. I know, that's a mouthful. We'll go through it slowly. For example, the following table is a summary of the rhino poaching incidents for South Africa from 2000 till 2010. So there's a table that they give us. They give us information for Northwest and for the Eastern Cape from 2000 to 2010. And they tell us how many rhinos were lost over those years in those provinces. Can you see? Northwest lost 63 and Eastern Cape lost 6. All right, so there was much more poaching happening in Northwest in that time. With what percentage did the poaching of rhino incidents increase in Northwest since 2008 to 2010 show all working? All right, now let's listen. Let's look at this question. With what percentage did the poaching of the rhinos increase in Northwest from 2008, so it's from this year to that year? That's what they want to know from us. They're not asking you how many rhinos were poached. They're asking you with how much percent did it increase. All right, so do you hear the difference? Now, what were we saying? We are saying you take the end value, and the end value for Northwest is that 44 in 2010, minus the begin value. The begin value is the 7 in 2008. And you divide that with what you have started with. And with what have you started? You have started with 7. And you must work what out? A percentage. And whenever you must work out percentage, it means that you must actually multiply with 100. That's how you work out percentage. All right. So now let's look how to calculate it. It's the end value minus the begin value divided by the beginning value multiplied by 100. Remember, in 2010, there was 44 rhinos poached. In 2008, 7 rhinos were poached. So that is our begin value. This is our end value. All right. And you divide that by the beginning value. And the beginning value was 7. So that is the same. And why do we multiply by 100? Because we want to know the percentage. And as you can see, it's very, very large. It increased by 529% because it was 7 that was poached in 2008, but in 2010, 44 was poached. So it's more than double, because if it was double, 100% increase, it would have been 14%. But like you can see, from 7 to 44 gives you an increase of 529%, which is a lot. And please, like I've said, this formula you will not find in the textbooks, but the examiners expect you to be able to calculate this. So it's very, very important. Remember, you want to know how many times it is more than your starting value. That is why you divide by your starting value. And that is what I said. All right. We've come to the end of this session of today's lesson. Thank you for watching this lesson. Please remember to sanitize, wear your mask, maintain social distancing, and keep safe. I see you again on Wednesday morning, 8 o'clock, when we will look at homeostasis in humans. Enjoy your day.